Okay guys, what we're going to do is I have a review for the lab practical, and this is for the zoology lab practical. So I want to make sure that you guys know it's going to cover using the scientific method, the microscope and cells, gametogenesis and embryology, and taxonomy and classification. Okay, so before I get started talking about the content of the test, remember that this is a hands-on test, but I do have what I call the regular questions, which are, maybe definitions or things like that, or questions that might have been in the lab exercise. That's not hands-on. But also remember that the test is going to be timed. You'll have 35 minutes to take the exam. I'm going to allow you to move through the test questions at your own pace as long as there's only one person per station. And if someone is at the station that you want to use, then you need to just wait patiently without hovering around them, or you need to just go, work on some other questions and then come back. But everybody will have the 35 minutes, and we're all going to start at the same time and finish at the same time. So you definitely don't want to be late, because then that will just eat into the time that you have to take the test. Okay, so for the content of the test, remember that I told you, like for example, in the using the scientific method lab exercise, this one I can't really test the hands-on part that we did for our experimentation in there, but I'm going to ask you like what I would call the regular questions about definitions, and definitely know the steps to the scientific method and know all of those important bolded terms that we had there. So definitely study that. And then the next one is about the microscope and cells. This one you definitely want to know about the microscope. Remember we learned about it, that it was monocular. It only has one ocular lens. And then some of the bolded terms in here are important, so you need to make sure that you know them. I'm going to ask about that. And then definitely you need to know about the parts of the microscope. I'm going to have, let's see, probably 1, 2, 3, 4, 5, 6, 7. I'm going to have 8 questions where you have to identify different parts of the microscope. So remember the way it'll be set up is there'll be a microscope and it'll have a label on there. So if it's question number 10, then you'll look for the label that has a number 10 on it, and then that's the question you'll answer. That's the part of the microscope that you would have to identify. Okay? And so then I also will have questions about the organelles, identifying the organelles on the animal cell model. That was in the lab exercise where you had to draw the animal cell model and then label the ribosome, cell membrane, Golgi body, cytoplasm, vacuole, centrosome, nucleolus, nucleus, mitochondria, endoplasmic reticulum, lysosome, and nucleus. Oh, I already said nucleus. Anyway, so this is the animal cell model in case you don't remember. And so I'm going to go through the parts with you. When we had the lab exercise, I had this that, let me see if I can hold it where you can see it, maybe. And it told the different parts. I don't think you can read it, but it tells the different parts that are on this model. And so remember that the white spots that it has on here, the white dots, those are representing ribosomes. They're going to be found mostly on the ER. So this area that you see here that's kind of this green and blue, that's going to be the endoplasmic reticulum. And I don't usually ask about the difference between the rough ER or the smooth ER, but just in case, remember the ones that have the ribosomes on them, the white dots on here, that will be the rough ER. And then where you see it just looks like dark blue and no ribosomes on it, that's going to be the smooth endoplasmic reticulum. And then let's see what else we need to know. The centrosome, remember, is this area here, and inside we have the 2 centrioles. the nucleus is this middle portion here, and then inside of the nucleus, remember we have the nucleolus. Let me see if I can hold this a little bit better where you can see it better. Whoops, well, I'm trying. And let's see what else. The Golgi apparatus is right here. It's kind of a pink color. This is the Golgi, or Golgi bodies. And then coming from the Golgi, you can see these, it looks like little bubbles. Those are vesicles. And then let's see, what else did you have to know? Well, you know just the liquid portion that would be inside of a cell. All of this, the liquid portion is the cytosol, but all of this in the cell, that's the cytoplasm. And then, let's see, what else did we need to know? Mitochondria, here, you need to know mitochondria. And then here, this would just be a vacuole. Okay, so there's a vacuole, and then let's see, what else did you need to know? The yellow ones, those are going to be the lysosomes, so you need to know that, lysosomes, anywhere that you see the yellow. If they're gray, they look similar, but if they're gray, those are going to be vacuoles. I think. And I think that was all you need to. Oh, the cell membrane is just, you know, the plasma membrane on the outside. Okay, that was everything. Okay. Right, and then I'm probably gonna have you identify, let's see, 1, 2, 3, 4 different organelles. Oh, be sure you know when we're using the high power, which adjustment knob is not okay to use on the high power. Do you remember? The coarse adjustment knob is not okay. You must use the fine adjustment knob when you're on high power. And do you remember what it's called whenever our microscope lenses are set up in a way that if you have something that's in focus on low power and you move over to high power, it's pretty much going to be in focus still. Do you remember what that's called? Parfocal. Okay. Okay, so then you also need to make sure that you study, like in the gametogenesis and embryology lab. Remember, you had to study the grasshopper testis lobe, and we saw the spermatogonia and all the various stages of development of the spermatozoa. So I'm definitely going to have a couple of questions about that, where you have to identify. So, like, for example, we have the spermatogonia. Okay, so make sure that when you're looking at the lobe, like I'll already have it set up on the microscope, make sure that you can orient so that you know, okay, up here, then that would be spermatogonia, and then down here you can actually see where the flagella of the spermatozoa would be developing, and so this is going to be the spermatozoa. Okay, so now I know, which direction it is, and so I'll ask you, I'm going to ask 2 of them to identify 2 of them, like spermatozoa or like spermatozoa, I'm sorry, down here, or like spermatogonia up here. And then we also had to know, remember we learned about the development of the sea star. So definitely make sure that you can identify the stages that you had to draw. So you had to do the unfertilized, the fertilized undivided egg cell or ovum. And remember, they look like they're just shaded in. Oh, I'm pointing at it and I'm not showing you, sorry. And then what was the next one we had to know? The morula, we needed to know about that one. So that one right here, kind of like a cluster of grapes. And then we had to know the blastula. Remember, that's, I don't know if you can see it very well there, but that's a hollow ball of cells. And then the gastrula, remember right here, you're going to see that it already has the blastopore and the archenteron. Remember, the blastopore is going to be the opening into the archenteron. The archenteron is going to be the tube. It's the primitive gut. So make sure that you can identify those as well. And then also, remember we had the summary where it talked about the development of the sea star, and you know it has radial cleavage, indeterminate development. The blastopore becomes the anus, and the second opening becomes the mouth. Remember those things, okay? I'm going to ask questions about that. Let's see. Oh, and then for the classification and taxonomy lab, remember that one, I'm going to ask you some definition type questions. So definitely know the bolded terms like dichotomous key. And then I had information about binomial nomenclature being the two-part naming system, and how sometimes we use trinomial nomenclature whenever there's a subspecies. And remember, a subspecies is when there's going to be geographical variation, or varieties or races of the species that exist. So, and do you remember that third part of the name is then called the subspecific epithet? Okay. And then lastly, I will have this key to the major animal taxa. I'm going to have 2 questions that go with this, and I'm going to have 2 specimens, and you're going to use that dichotomous key to figure out the class and or the phylum of those 2 specimens. And I think if you study all that, I think you'll do really well on the lab practical, but be sure to let me know if you have any questions about anything before Monday. I may not be able to answer except for right before, because I won't check my email over the weekend, but you could email me and then check it on Monday morning. Okay. All right, thanks.